practical session board. So be very strict on the attendance at 10 o'clock and allow only these people to attend throughout the day. If someone is coming, ask me to meet me, and then I see what reason they are doing. So, okay, I hope we will attend it seriously. I again invite Professor Chodoro this session, lecture 1 and lecture 2. After the lecture, we will have a short and then second lecture. Professor Chodoro is yours.
Okay, good morning, everyone. So you managed to rest a little bit? Yeah, I hope. Well, we're going to continue with the story and trying to go to a little bit more details on what we were doing actually yesterday. So if you think about yesterday the work, what we were doing in the practical, he's explaining the procedure again, and then maybe some doubts that you have will be fixed better. Okay, first of all, we have the commercial samples. We had this pickles, we have the duck heap, and we was. Can you see the plate in the bowl? You have the bottom up. Then we we homogenize the samples. So we get the liquid samples, so we don't need to homogenize. We scratch from the mango to make it easily. And since we're not caring about the number of the cells we have in the samples, we was directly spreading with the loop. So we're putting the samples in the top, then we cover with a second layer of the agar, and the plates are incubating now. I hope will be some individual colonies today, and we're going to apply a third level that will be here. We're going to cover with another layer of the agar, so we want you to see something like that today. This is what we want to see, because this means this colony here, this one most probably is producing something, and not this one, because if this one, the zone will be like that. So something what we want to see. I hope we will see, because no one can guarantee, but this is the point. So again, here it's explanation of the entire process again. You have the sample, sorry it's in Portuguese, but I'm explaining it now. Your samples you can you can mix with the physiological water always. Then you can make a dilution, then you can plate on the agar, then you cover with another layers, then you incubate, then you observe from the incubation zones, if you have. Then you pick one colony from the incubation zone, you go to the culturing them in the liquid cultures, and then you're going to check catalase, gram coloration, and then some other test, but it's coming from the way as well. There's another way. This is the way when you try to explore the new producer, if you have the raw product. In case you have the real culture, already you have your culture collection in the laboratory, and you want to explore if some of the culture collection is already going. So you do it much more simple. You make agar with the test organisms, and you spot just a small amount, could be 10 microliters, could be just one touch with the loop of the culture, pure culture that you want to test. You incubate and you observe. If you have something like that, the culture point and have the incubation zone, there is a chance. But this is only in case if you have already a pure culture. So you're going to observe something like that, I hope, tomorrow, because after you see the test organism is growing, and here most probably it's a bacterium. Very important point on this experiment is to go to have the pure culture, the standard microbiological procedure. You need to be sure that you have the pure culture by streaking on the plate, picking a colony, streaking again. It's taking time, but there's no another option, because if you have contamination, you can never work properly. And then freeze dry, if it's possible, or keep in -8. It's always important to keep more than one Eppendorf, because you're going to keep it in -8 in the presence of glycerol as a career protector. But it's important to keep at least 5 or 6 cubes, because sometimes happens something. Even it's good to put it in the 2 different freezers, in 2 different places. I was working in South Africa, and one laboratory took fire. Everything was on fire. and they lost entire collection. This is the good point. Very often what we do, we keep it in our another building laboratory freezer. We're just taking one freezer to a different building and we keep all the collection in duplicate. Because if you lose everything, imagine you're doing a PhD on the last year of the PhD and you lose the collection, you lose the new bacteria. All your PhD is gone. You cannot defend because you cannot continue working. You need to start from scratch. But then it's important to check if this bacteria is really producing a bacteriocin or not. Because as remember from yesterday, this could be an acid, this could be diacetyl, this can be a Reuterin, this could be even an antibiotic if it's some strain of bacillus that's producing antibiotics. So we need to do the check for the bacteria. Normally the check is quite simple. We need to have a supernatant, so you centrifuge, you obtain the supernatant. Then you need to correct the pH. Why? Because correcting the pH, you're excluding production of the acids. In this way, you're sure that what you will detect as activity not going to be acid. And we need to do the treatment for 10 minutes at 80 degrees Celsius because in this way, you can destroy, if it's any hydrogen peroxide produced, you will destroy it. And as well, if you have any sensitive enzymes, proteinase, you will destroy them. Because some of the lactic acid bacteria, they're not very typical. They're not good producer of proteinases, but some of them produce. Imagine you have the bacteriocin, it's a protein, and you have close to him a proteinase enzyme, what will happen? Enzyme will destroy the bacteriocin. And then we spot 10 microliters of the surface of the plate and incubate and observe for such an activity. Now it's an entire method explaining more in detail. So you culture your bacteria. You can incubate, well, in this case we put 30 degrees, but if you work on the strain that you want to use as a probiotic, you need to pay attention what kind of probiotic are you going to use. If you're going to use it for human, are you going to use for the chickens, are you going to use for the fish? Okay, someone veterinarian here? No one with veterinarian background? What's the temperature of the chicken? Yes. Please, just try it. Are they warmer than humans or they're colder? Well, they're about 39, 40. So if you develop the probiotic for the chickens, you need to be culturing every time in 40 degrees because this bacteria want to know how it behaves when it's in the body of the chicken. And if you're doing a probiotic for the fish, it's any fish living at 40 degrees Celsius? No. It's like 25 maybe, but even lower. Depends where the fish will be growing. If it's in the Amazon, in the tropical area, obviously maybe 30, maybe not really, maybe 25, 30. But if you have the salmon in the Norwegian area, it will be like maybe 15. So you need to check the temperature that is objective of your work. You grow your centrifuge, you obtain the free supernatant, you can correct the pH, you do the treatment to destroy the proteins and as well destroy the hydrogen peroxide. And then you need to spot on the plate. This plate, when you use for the bacteriocin test, we prefer preparing them with a soft agar. So in this case, since I work with Listeria monocytogenes, I'm using the BHI with the agar, and then I inseminate Listeria inside. Normally, we need to go to the level of 10 to 5, 10 to 6. So there is a practical tip how we can do this. It's more or less 1 microliter of Listeria culture for 1 milliliter of the agar. And in this way, you're reducing already 3 logs down. So later today, when we go to practical, we need to pour this third layer on the plate. So we're going to do these dilutions. And it's a very practical way because, okay, if you prepare 1 plate, you can make a 100-time dilution. You put 1 milliliter of this diluted Listeria, then you pour in the top agar, you mix the plate, it's fine. But if you need to make 50 plates or 60 plates, it's not easy. So rather make this a proportion, 1 microliter of the overnight culture of the test microorganisms with for each 1 milliliter of the agar, you mix in the bottle, you mix the bottle, you pour the plates. And then you sure that you have the same amount of the Listeria for all the plates. Then you spot 10 microliters of the supernatant in the top, already treated. Then incubate and observe for the presence of the cells. And if you have the zones, bingo, it's working. If it's no zones, let's start from the beginning. We need to look for another strain that can be used. Okay, now the question is, how strong is our bacteriocin? The way of measuring the activity of the bacteriocin is the same way that people measure activity of the antibiotics and the enzymes in the units. So for the determination of the units, we're using critical dilution method. So now it's going a little bit more complicated because I need to explain this method. So you're taking normally the ELISA plate to do the dilutions because it's easy. You work with a small volume, and you can do it in the Bendorfs as well. So you distribute to each of these places a phosphate buffer because we need to be sure that the protein will feel the stable at the presence of the environment. Normally the proteins are very well having good stability in the phosphate buffer with pH 6.5. Could be 25 up to 100 millimolar, no problem. It will be okay. So you're putting like the same volume here. Let's say you can put 100 microliters for all of them. Then you take your bacteriocin, your supernatant, and you're putting the same volume. If you put it 100 microliters, you take 100 microliter of the sulfur supernatant. Mix, pass 100 here. Mix, pass to the next. Mix, pass to the next. So you're doing 2-time dilutions, serial dilutions, and then from each dilution you will spot on the plate, and then you can observe. And to calculate the activity, actually we need to take consideration which one of the spotted material don't generate inhibition zone. So this one will be our critical dilution, and as the inhibition zone, normally believe that it's a 2 millimeters diameter. If it's less than 2 millimeters, better consider it as a negative. So one arbitrary units, it's actually calculated by the dilution, because we made it 2 times serial dilutions, and it's a number of the first dilution that's not presenting. In this case, we can consider it, let's say, 7, and P, it's a volume what we deposit on the plate in microliters. Because remember, we deposit, where is somewhere it's written, yeah, it's here, but you cannot see it. 10 microliters we deposit on each place. But well, the result is in milliliters, so we have this P like a conversion factor, 1,000, to have from microliters to milliliters. So now please take a piece of paper and let's calculate. Let's make practical things. Okay, let's, we can see like half of the cell, half of the room. We can consider that here it's no inhibition zone in number 4. Finish, no zones here anymore. So one of the group can calculate with number 4, another group can calculate it with number 7. So you're having dilution. It's 2-time dilutions on the exposition 4 or 7, multiplied by 1,000 to have conversion from microliters to milliliters. How much will be the activity? How much?
Volume 10 microliters.
Volume is 10 microliters.
3,000.
Okay? And another group that is 7, who calculate with 7?
1,600.
But you're doing PhD, it's easy for you. Right? Well, you understand the logic. Okay, now let's make it more difficult. If you have the very strong bacteriocins, obviously you can have it even inhibition zone here, attained. So you have 2 options to make more dilutions. You can dilute up to 16 times, or you can do it different ways. You don't make a 2 times dilution, you make a 10 times dilutions here. So in this way, you need to take attention that D will be 10, not going to be 2, and then you can have your results as well. This is very important because if you just do the inhibitions on the diameter, can be big variations. And this is a problem because you pour a little bit more agar, you pour less agar. Your agar was 2%, your agar is 1%, your agar is 0.8. All this can interfere in the inhibition zone in diameter. So very often it can give a wrong result. So making a critical dilution, you can always have a better result that you can compare with the other one. So now remember, when we was talking about the bacteriocin, we mentioned that they are proteins. and how you can actually confirm that really our substance that we have activity, it's really bacterial. So first point, we need to do to show that it's a problem. Okay, if you treat with proteolytic enzymes, very clearly proteolytic enzymes will destroy the activity, and you will know activity after exposure to proteolytic enzymes. So you can take any proteolytic enzymes that you have in the lab. Don't need to be specific list. You can check what left over from another project, what is available. Now, fact that bacteria can produce the lactic acid, you need to be always sure that you do the neutralization of supernatant to avoid the fact that is some lactic acids produced. But for the industry, it's perfect because in the industry, you tell them, listen, you have salmonella in your sample. It's already enough for them to have a big problem in the lab. Be a very kind of general approach, but not really in detail. Imagine how we can distinguish different strains on the same species just on the protein level. This will be very difficult. If you think about Lactobacillus plantarum, Paraplantarum, and Ventosus, they're difficult to be distinguished on genetical level sometimes, and only on the protein level you can make this work. Not really. Well, now the top not going to be happy with my comments because they want to sell their machines. But in the same time, now the top, the companies, actually Bruker, Biomer, or the rest of the companies producing this equipment, they went for the new generation because they realized that their equipment, in fact, is not so sophisticated to meet the scientific exigence. They meet industry, but not on the academic level. So they made it a new addendum to the machine that they have the Fourier transform infrared spectroscopy. So once they have the classical mounted off, they have the spectroscopy second section that actually it's helping to do better distinguish, and including then some of them have even a Rabotyper that helping to really find what do we have. Only the small problem, this one cost $2 million. Well, even in the very healthy labs, this is a lot of money. But this is the same time, I think it's some strategy that several universities have. They have these multi-user facilities. They buying equipment for entire university. Sometimes even two universities buying the same equipment to use it by two of them. Because this kind of equipment, such a price, I mean, you need to combine with your neighbors. You cannot just have it in your lab. You need to do it as a central facilities of the university. They will serve several faculties, and this way it's work. Because this kind of equipment, if it's work, it's okay. If it's standing, stopping in the corner, it don't generate nothing. It needs to work. And in my lab, I'm not going to do the identification every week. I'm doing like 2-3 times per year when I have new collection, new strain isolated, and they need to do so. Obviously, the central analytical facilities is a good option for this. But it's something that is work quite quick, quite easy. But again, even with this identification, I will go for the full genome sequence. Because these days, full genome sequence become a compulsory point as well for the identification. If you want to use the strain as a probiotic, you're going to use as a starter culture, you need to do the full genome sequence. There is no option. Several agencies, to order to certificate your product, they ask you to provide full genome sequence. They want to be sure that you have sequenced your DNA total, you made it appropriate bioinformatic analysis, and you show that the strain don't have the dangerous parts in the genome, because again, safety is priority. You don't want to introduce something that having genes that will be related with some dangerous feature. Well, now you know that you have the bacteriocin, you know the identity of your bacteria, and you need to think about how you can characterize your bacteriocin. First of all, you need to be sure at what level your bacteriocin is produced and where you can stop the fermentation. Okay, in the laboratory, if we do the 24 hours, it's not a big deal. But in the industry, if you can stop the fermentation earlier, it's mean money because Imagine in the lab we use the lab tube like 10 milliliters, so we're using the Erlenmeyer flask with 100 milliliters, but in heater steam there will be 20,000 liters. You don't want to heat this huge bioreactor for longer period. So obviously you can do a very simple way. You can do the growth production study. So you just grow your culture. You're taking every hour samples and you measure optical density because it's easy to just to do the plating and do the cell count. But in certain extent, the optical density gives you a good idea about the growth. So you grow your culture. You see it's reaching the stationary phase more or less 16, 70 hours, even 15, and they stay in stationary phase. In other cases, reaching a little bit earlier, about 10 hours or 11 hours, reaching the stationary phase and go there. Then we do the acidification as well, because on this way we can understand something about the structure of bacteria. Because sometimes bacteria, when they go to the low pH, they can make a clumps. They go together. Two of them, they will, two molecules of the protein, they go together, they go four. And very often when they make these clumps, they are not anymore active. They're losing their activity. But this is normally associated with low pH. So it's important to know how the pH acidification going, and as well very important to know what's the level of the bacteria in production. So in this way, you can see we was doing a fermentation for 48 hours, for 2 days, but in fact, after, let's say, how much is it, 21, it's staying stable. Why you should go for 48, even in 21 I can stop it? Don't make sense, right? Okay, for the lab, if you put it on Friday and you want to work on Monday, it's already too late because it's going down. But I mean, you made it a culture in the let's say in the 8 o'clock in the morning, but then you had a meeting and you stop at the more lunchtime, okay, because you're in this area that you already have the maximum of production. But again, you need to know where is the maximum so you can stop it. In the second case, in the bottom, you see have a very big peak in the end. But this makes sense to have this high activity because activity here as well is high because it's related for like this will be 2 with 8 zeros in the back activity. It's quite high. You have more here, 1 with 9 zero. So 1 zero more, 1 log more of activity really make a difference, but make difference on the energy use and the time. So I can rather stop again on 21, 22 hours here because I have enough and you don't need to go too much. And here as well, you risk to go down because making these clumps. Then is another problem. Okay, here it's showing like stable optical density, but could be a little bit kind of not true. Because obviously here you have dead and live and dead cells. In stationary phase, you have live cells, you have dead cells. The problem with optical density, you're not detecting live or dead. But if you do the plate count, you can see that actually start to declining here. If you do the cell count, not just optical density. And when you have too much dead cells, what's happened? That cell is lysing, and they liberate proteases from the inside. So some proteases could be available in the end of the process, in the end of the stationary phase, and they can interfere with the integrity of your bacteria. Then you know what is the time, you know what, how long you need to take a fermentation to have the optimum bacterial production. So you know where you can stop the process. Again, this experiment is done in 37. But if you work with a probiotic for different kinds of targets, poultry or fish or even agriculture, you need to adapt to the temperature that is appropriate. But now is coming another problem. Lactic acid bacteria, what is their growth temperature preferences? More or less, Enterococci below 15, they don't like it cold temperature. Lactobacilli below 20, it's not so easy. Going up more than Streptococcus thermophilus, 43, 44. But if you go higher, they will be not happy. I mean, they have their preferences, and we need to respect their physiological needs. So now, you're going to do something for the crabs. Crabs, normally they grow in the very cold water. So obviously, lactic acid bacteria not going to be a good potential probiotic for the crops because you need something that will be happy in cold temperature. So you need to look for the different group of microorganisms. And this could be archaea, this could be some bacillus, because bacillus they have larger temperature preferences. So again, lactic acid bacteria cannot solve all the problems because again, you go to the living organism, and this living organism has specific temperature, and lactic acid bacteria has specific preference for the temperature. So we need to match together in order to work. But again, we need to find now how this bacteriocin is stable. Because if it's not stable, we're not going to work. Imagine you having bacteriocin that is not stable in low pH. Can you use it in dahi? Dahi, what's the pH of the dahi more or less? I presume it's similar like to this on the yogurt, presume. It will be like 4 something, called 3.9, 4 point something more or less in this area. But if the bacteriocin is not stable at 4, they're not going to work in this kind of product. So you need to check the effect of pH. You just take a supernatant. Please, when it needs to stop, you can let me know. Okay, because in Brazil, normally I'm used to do it lectures. We're having 4-hour lectures. We're doing everything in one time. But again, since Santos promised some tea in the break, you can let me know more or less 5 minutes before, and we can make a break. Okay? Deal? He's not here. You can tell what you want against your body. But he may be watching on the video transmission. So we're supposed to keep good. Okay. Then you take a supernatant, you expose to the different pH, you keep for 1 hour, then you need to correct pH back to neutral, because to be sure that you're not detecting acid, right? And then you check activity. As well, you can check the effect of temperature. Now we're not talking about the growing microorganisms of different temperature. We're talking effect of these temperatures on the stability of bacteriocin. This meaning you need to produce your bacteriocin, you have the supernatant, and then you put on all these different temperatures. Keeping for 1 hour or 2 hours, then you're taking out and you check the activity. Important thing, it's 121. Why? Because if you're going to apply the bacteriocin in some medical application, you need to be sure that it's sterile, right? You need to be sterilized the bacteriocin. If you're losing the activity, obviously this will be a problem. Well, we can work on different ways to sterilize filtration or whatever. There's several methods, not only temperature. But again, it's important, but it's a good point because bacteriocin are smaller than 10 kilodalton, majority of them. And they have this stability on the high temperature. Enzymes, they're much more bigger and high temperature destroy their activity. So some of the bacteriocins could be bigger than 10 kilodalton, this is class 3, and they could be maybe losing their activity when exposed to the high temperature. We need to check effect of ox bile, sodium chloride, food additives, medicaments on the stability of bacteriocins because you're going to use your bacteriocin for biopreservation of, let's say, cheese. Cheese is having a high level of sodium chloride, having presence of milk. So you need to check effect of milk. You can use milk powder or you can use sodium chloride and check to see if these components making problem on the stability of the bacteriocins. Problem with the sodium chloride is because he can act like taking water, and once he is dehydrating the protein, maybe in the biggest protein, can have some negative effect. Regarding why milk could be destroying the activity of the bacteriocins, the simple fact milk is several proteins, caseins, etc. Proteins, and these proteins can make a complex with the bacteriocin and can deactivate them. But you need to check this on the plate because only in this way you know future you can apply it in these products. What is the point to make a cheese production? You're applying the bacteriocin only after they discover that they cannot work in combination with the milk proteins. So rather check first on the plate to save you time later. Some of the components, surfactants, etc., is important to know if they're having this effect because very often we have them in the purification processes. Obviously, no one will eat SDS. It's obvious. But SDS is used for the purification processes in the determination of the molecular size. So if we know that the SDS is doing a negative effect on the stability of the bacteriocin, obviously you're not going to use SDS page to do the molecular size of your bacteriocin. You already know that this method is not appropriate. Twin 80 is a part of the MRS media that is used for the production. And in some cases, it's a problem because surfactants can have negative effect. And then you know you need to make MRS media without twin. Okay, we'll make some foam when you do the mixing in the bioreactor, but again, this twin, if have the negative effect, we need to take it down. Triton is used in the purification steps. You're already checking on the plate and you know what kind of purification later you can do. Then coming a question to the activity of the bacteriocins. Normally, when we check the activity, we need to know what's the activity against closely related microorganisms, because according to the definition, bacteriocins are active against closely related. But then imported human and foodborne pathogens, because we want to know, we want to apply this bacteriocin. If you have the bacteriocin that kill another probiotic, it's not really making sense. But if you kill listeria, if you kill Staphylococcus aureus, this is a good point because you have something that works against the foodborne or human pathogens. But in the same time, you want the bacteriocin to be not active against other starter cultures. So you need to check different lactic acid bacteria from the same ecological niche. Okay, yogurt. Remember yesterday I was telling you, yogurt is starter culture, always Streptococcus thermophilus and Lactobacillus delbrutis of species bulgaricus. If my bacteriocin killing one of them, I'm going to apply it in the yogurt? No way, because I will destroy the starter cultures. And then is coming this additional, when you check the activity against gram-negative, against viruses, mycobacterium, yeast, and fungi, because this unusual activity, considered actually unusual 20 years ago, these days become more and more interesting because several strains been shown that they have this activity, and this can be used. Now, you having, you know what you can kill, you know that you have the bacteria, you need to know, are you really killing or you having just inhibition? It's very simple test because you can grow, you test organisms. Okay, listen, I like Listeria monocytogenes as test, and we'll use it as an example most of the time. So you grow the Listeria monocytogenes, and you add a self-free supernatant containing the bacteriocin, more or less in the early exponential phase. So I added after 3 hours. In another experiment, I added in the middle of the exponential phase, around 7 hours. And I was just simply following changes in the optical density to see what's going. Okay, Listeria without is growing very well. But Listeria, when it's added the bacteriocin, is keeping the same level. Well, the question, are these Listeria just inhibited or killed? By the optical density, you cannot say. But again, doing all this experiment with counting, plate count all the time, you will run out of the lab and you don't want to go anymore back to this laboratory because it's a lot of work. Every hour you need to make a dilutions, you need to plate. It's taking a lot of work. And by the simple measuring of the optical density, you can actually see what's going on. But then it's important here in the end, let's say on 12 hours, you take a sample, you make a plate count. It's 1 or 2 points you need to do it, but it's not 14 times, it's only 1 or 2. You see, I'm trying to save the money, save the planet because we're not going to use too much plastics, and as well save my time. In fact, there is something interesting opinion. They say the scientists are lazy people. And they make a good discovery because they don't want to work too much and they find a way to have the good results with the less work. And actually it's true. You're using your brain, the way to do it less, work with my hands, but to have more results with less work. So actually it's a good point. And then when you do the cell count here, if you still have the number of viable cells, it means you only have a bacteriostatic. Listeria is not happy, but he is not dead. And if you don't detect any viable cells, this is a good point because your bacteriocin most probably is bactericidal. You can always keep this culture late and go like there, and you do it in 28 hours or whatever, 30 hours, and you can see because sometimes they starting to recover. In this case, when it's bacterial static, since bacteriocin is finished, bacteriocin is not forever, and is finished, is already used to block the receptors, etc., to inhibit the listeria. In some moment when it's not anymore, they can maybe start recovering. But again, this is additional. But think about antibiotics you're taking only once and never again. You're taking every 8 hours, every 12 hours. Okay, so more sophisticated, you take it once per day, but you need to give your body constant time for like at least 5, 6 days antibiotic to be sure that you really destroy the pathogens. Same thing with the bacteriocins. They, in some cases, you can make this experiment more sophisticated, and then you add again after 12 hours, you can add here and see if really will keep the and eliminate the pathogens. But again, this previous experiment was showing you in the growing condition. So we can actually calculate the cell lysis using a non-growing condition when you have the cell suspension, and you can treat these cell suspensions with some levels of different level of bacteriocins, 12,000 units up to 200 units, and then you check the changes, and you can do it in actually an ELISA plate on the microplate reader, and you're checking the change of the DO on the microplate reader, but he's not growing condition, so it's important. And then you can see actually that even the small levels of the bacteriocin have almost the same effect on listeria. 80% of the cells being lysed in the presence, even on 200 units per milliliter, almost the same results with 12,800 units. And over the time, you see in the beginning, after 3 hours, you have reasonable, like 30% lyse of the listeria. After the 9 hours, it's growing to 80%, and after 18 hours, it's actually almost 85%. So you can have more sophisticated methods. If someone will work with the bacteriocin, I'm happy to sharing these methods with you. It's a simple, not a difficult, and giving a good information. What is the quantity of the bacteriocin that you can use, and how the process is going over the time. Remember when I was telling you to check the SDS, how affecting the stability of the bacteriocins, and this is important to make this SDS page, determination of approximative molecular size of the bacteriocin. Some already one made the SDS page for the proteins? No one worked with the proteins? Okay. SDS page is a very good method because in this you're generating good gel, and you're having a separation of the proteins because of their molecular size. The problem, you need to make a gel into you make several repetitions of the use samples, and then you need to cut the gel, and one of part of the gel will go with the staining, which could be Comasi blue, could be silver staining. Silver staining is much more sensitive. Comasi blue is less sensitive, but it's enough to see. This is a marker, different already known size of the proteins. And then you need to, now, please, who know where to stop the light in the front? To see the inhibition zone, we need to stop the light. So know where to stop it? But okay, if no, I can show you again. Can you see there is an inhibition zone here? Can you see the difference? Because it needs to be a little bit darker to be observed easily. So one part of the gel, this is the marker that I was using. This is my protein, semi-purified protein that I put on the gel. So you can see it's like a band here, protein band. I don't know if it's very well visible. With silver staining, it will be much more better, but the Comasi blue is cheaper. Silver staining is more expensive. You know, we need to save money, no option. We don't want to waste money. Sometime in the first screening, we'll do with the Comasi. When it's giving a good result, we can repeat later with the silver staining. But the silver staining could like 100 times more. And if imagine the students start doing something, making error and another error and another error and spending expensive material, I want to prefer to do it the cheaper way. And after that, doing with the more expensive. So then when you stain, you see something here, but you don't know. Maybe it's this protein, maybe it's this, maybe it's this. You don't know which one. This is why you cut the gel in two halves and using one half of the gel that is having again same material being running here as well. And you just wash the gel with the specific protocol, put on the plate and cover with the agar with the test organisms. And this showing inhibition is only showing where is the band of the protein band of interest. It is there. So then you compare the two parts of the gels, you put them together, and you say, okay, my bacteriocin most probably is this band, that is corresponding something about 4 kilodaltons. It's not a difficult way. It's taking time, it's taking work, uh, needs some experience to manipulate, but once you're comfortable, it's going really easy and not a big problem. Well, this is approximative way. The problem with protein gels, you have very approximately because it's based on the migration of the protein, based on your eyes to observe and to analyze. So we can go for the different way to determine the size of our protein and as well to determine the amino acid sequence. I need to, in 5 minutes? Okay. So now we need to make our protein. We need to know what is the sequence of the protein. This is the real characterization of this protein. So we have two ways to do it. Easy way, PCR. So you look on the literature what is available as information for Lactobacillus plantarum or for Pediococcus pentosus, which kinds of bacteria was described. So you can buy the primers based on the literature, you make a PCR and you start shooting. If you have the positive results, great. If you don't have positive results, need to look further. But again, my bacteria may be producing a completely new bacteriocin, so we need to go further. So based on the amino acid sequence of the purified bacteriocins, we can generate the primers based on the sequence of the protein, and then we can do another PCR and detect the allergen. There will be some examples on this, don't worry. Then another question, will be our bacteriocins on the plasmid or is on the genome, on the bacterial chromosome? Another question, in the beginning, they believed that the bacteriocin always will be on the plasmid. But then appearing that is not true. Some strains don't have nothing, no plasmid DNA, but they produce it. So obviously bacteriocin genes could be on the plasmid, could be a bacterial chromosome, no difference, no, could be one or another one. So if you have the plasmid, you can start doing the curing the plasmid, eliminate the plasmid, and checking again for activity. And the exclusion approach, you can see, okay, I'm losing activity, but I lose the plasmid. Most probably this plasmid is supposed to be. In this case, easy way, because the plasmid is not too difficult to sequence. You sequence the plasmid, you analyze the sequence, and you have already your sequence of the based on the gene, you can reconstruct the protein, etc. But when it's not, there is a question. Okay, you can detect by the primers. You take, you say you're having Pediococcus pentosus cells or Celiac lactis here, any Pediococcus, and you detect the presence of Pediocin PA1. Okay, you can be happy, you can be sad. You detected the same bacteriocin in a different strain. Well, let's do the PCR. Let's have the PCR product, sequence the PCR product, and analyze this sequence. Maybe will appearing that you have a mutation, that is already something different. Because you blast the sequence and you detect, okay, I have Pediocin PA1. But this Pediocin PA1, it's the same like this one in the literature. Now, coming to this point, you can have the same, even same genetic sequence. But remember from the basic on the genetics, the triplet for the same amino acid. You can have it, let's say, okay, let's take one. Serine. Serine can be UCU, can be UCC. There could be a different triplets pointing the presence of same amino acid. So based on actually this point, we can come into a very important point. Okay, everyone agree this is an art? And someone don't like it, Mona Lisa? Purification of the protein is art as well. Because when you purify the proteins, you don't have a recipe. There is no book that will tell you to do this. There is a combination of several approaches, and based on each step, you will decide what step you go further. So this is the point because again, okay, we can stop here. This is the point because on the DNA level, you detected some differences, but because of the different triplets going for the same amino acid, maybe you have the same amino acid sequence. So the DNA level could be some difference, but on the protein level could be the same. And the opposite. If you start from the proteins, you can detect that you have the gene differences. But already gene differences in the structural gene of the bacteriocin is showing some that you have the different bacteriocin. No, it's the same bacteriocin, but by the different gene. Then it's again giving uniqueness to your strain. And if you want to patent, this is a good argument. Okay, I have producing the same bacteriocin, but my strain is a different genetic organization. So it's making difference. Okay, let's have a tea, coffee, whatever you want, and we can go back in some time. I don't know I have no idea. What is the break? 30 minutes or? Okay. Please let's go to the coffee, tea, just references. Can we continue?
Yes.
You already had the tea? Okay, question. Who liked the coffee? No one. Okay, good people. Well, my preference is, well, I drink both of them. But the coffee actually is good. Good coffee, start today is good. Okay, let's go now on the purification problems. Because as I mentioned, purification is a real art. You need to combine the different methods because only this way you can reach what you want. Okay, if you have the protein that you already know the structure, you know everything about proteins is, but remember you discovering the new bacteria, and you need to do appropriate way of purification. Well, we have some recipes, but how we can apply these recipes together, it's not an easy task because you need to think about what after what, how, which condition, all this. But in general, some precipitation in the purification, starting with precipitation with ammonium sulfate. It's a very classical method, classical approach in the purification of the protein, because the ammonium sulfate can um, dehydrate the proteins, can make a complex with them, and can precipitate them. And only the important thing you need to play in the condition, of course, because depends on the size of the protein, depends on its cationic, anionic, etc. You need to use less or more ammonium sulfate. And the point ammonium sulfate will precipitate all the proteins. So obviously you're not going to make 90% ammonium sulfate precipitation because it will precipitate everything. You want to precipitate the bacteriocins, these specific proteins, but you don't want to precipitate other proteins. So you need to make a kind of game in the beginning, optimization. You're doing 40, 60, 80% ammonium sulfate precipitation. And then you check if a protein of interest is in the pellet or it's in the supernatant. Well, time consuming, but then you're optimizing your process. You can do the precipitation with the ethanol. Work very well with the RNA, DNA, but as well with some proteins. You can use in several cases high level alcohols. You can use the isopropanol, hexanol, etc. Because in this way you can attack some proteins, and not all of them. We can do the triton extraction. Remember, I was telling you to check if triton X114 in fact interact with the bacteriocin, because if it's not negative interaction, you can use the triton for the extract some bacteriocin because they're making this interaction with the protein, and you can take out of the system and facilitate your precipitation. This method been very well implemented in purification of one specific bacteriocins. So now you having precipitated, you made it ammonium sulfate. You can even use the sodium chloride precipitation. It's a little bit more rough, but okay, could be an option. You can made it to precipitate, and then you can use normally setback C18 hydrophobic based chromatography. Good part of this chromatography that is you don't need the HPLC, you don't need the equipment. You just have a small cartridge that is not very big, and use the syringe. And you using syringe and the cartridge to separate your bacteriocins based on the hydrophobicity, because most of the bacteriocins are hydrophobic molecules. And using this hydrophobic column and using appropriate solvents of isopropanol, you can actually make a fractions that your bacteriocin will be in this specific fraction, because you will do the gradient of the isopropanol, let's say 20, 40, 60, 80%. And you can actually go rid of other proteins and having a fraction completely concentrated with your bacteriocin. Another important thing when you do the ammonium sulfate precipitation, you need to go rid of ammonium sulfate. You can do dialysis, it's not that difficult, but you lose some of the bacteriocin during the dialysis as well. So using the setback hydrophobic column, when you're loading your material, actually in the first fraction, it's all the ammonium sulfate, because ammonium sulfate will not interact with the matrix of the column and will pass to the column, and you have it rid of all the ammonium sulfate already in the first step. Then, but setback still is not pure, it's semi-purified. You can use this material in several experiments when you need to semi-purify the bacteriocin. But if you need to go further, you need to use HPLC or FPLC or after purifier, and you need to be sure that you have pure material to go for next step for mass spectrometry and amino acid sequence. There will be some examples of this as well. When this is the more or less this is the basic principle way, but we can use some imagination and we can make some different approaches. Was observed that some of the bacteria they can stick to the cell surface of the producer cells. They stay there. They don't kill their own cells, own producers, but they can stay on the surface of the cells. So what we can do? We produce the bacteriocin. Bacteriocin is on the surface of the producer cells. Then we play with the pH. We put the pH very low on the cells only, because you separate cells on the supernatant, but bacteriocin is on the cells. You resolve in the buffer with a low pH, maybe 2, and then bacteriocin will be liberated because of the low pH. Then you centrifuge again, you have the supernatant, you correct the pH, and you have a kind of a concentrated bacteriocins on the present. This method was proposed very long time ago in 92, but still it's one of the good methods for quickly obtaining semi-purified bacteriocin. Only the problem, only few bacteriocin stay attached to the surface of the producer. Most of them, they're going in the supernatant. So you check if it's this case, great. If it's not, you need to look for another way. There was another method. When I was developing some probiotics for for woman health from a vaginal application, they observed that in this condition, cells can be in non-metabolitic but a very healthy condition for the cells, for the bacteriocin producers, and then they can actually export bacteriocin that is already produced inside the cells. So there was researchers, they generated a media, it's the description of the media here, that is simulating vaginal fluids. And then they putting these strains in this artificial environment. The bacteria, it's feeling comfortable, not growing, but continue expressing the bacteriocin. And since it's a very low protein environment, all the proteins that you detect, in the majority of them will be eubacteriocin. And then it's much more easy to go further and to make their purification. In the standard way of purifying the bacteriocins, normally, as I mentioned, you can grow them, then you can use ammonium sulfate precipitation to go rid of proteins that you don't want. But as well, in the beginning, we can use the Minitam system. It's a very simple, actually, filtration system that using filters of 6 to 8 kilodalton. So when you filter by 6 to 8 kilodalton, you go rid of all big proteins. But the bacteriocin is normally at 3, 4, 5 kilodalton, so bacteriocin will pass via this filter using in Minitams. Minitam is just commercial name, but in fact it's a filter, simple filter that can do it with a high pressure and specificity of the equipment. Then you do the ammonium sulfate precipitation, then you do the Septac C8. It looks like that this column. It's size of 2 centimeters. It's a very small. Then you can do the cation exchange chromatography, and you need to choose cation or anion exchange. Well, most of the time will be cation exchange because they are cationic proteins. Some of them, few bacteriocin can be anionic. And then you see something like that with the appropriate gradient of the solvents. And this thing is our bacteriocin. Well, the bank news, when you do this kind of job, you need to have the very symmetric peak. And this one is not symmetric, you will have like a second peak here. But we was quite lucky because of the gradient is having this kind of fake non-symmetricity. And when we do the mass spectrometry, we're having almost pure material. Since confirmed the mass spectrometry that it's a pure material, you can go for the amino acid sequence. But if this is not confirmed, you don't go for the sequence because obviously you're not going to have a good result. In another method, we was producing in MRS broth our bacteriocin, do the ammonium sulfate precipitation, set back, we obtain the fraction that was on the interest, and we put it on the size exclusion chromatography. By the way, Santosh will talk, we have a practical with you on the methods, chromatography methods. I think also on Thursday, right? Because tomorrow he's in Delhi, so on Thursday he'll make a practical with you. But in this case, we use the size exclusion chromatography, that chromatography based on the size of the molecules that you want to separate. And we was having to have, well, the problem in this way, you're having a pure peak. Okay, good. Well, first we have this one. When we put the material from the setback, you see it's like a mountains. Very often, not the biggest peak will be the owl material. In this case, was this one that is here. So what you need to do, every time it's going something out of the chromatographer, you need to collect and you need to check for activity to know which one is. Because only way to know which fraction that is interesting for you is to check the activity. There is no another option. So you do this chromatography 5, 6, 7 times. The good news is about 1.5 hours. It's not too long. But still, it takes time to stabilize the column, take time to run everything. So it's time consuming. You repeat several times, you collect this peak that is of interest, you put them together, you do the ray injection of the same condition, and you have this beautiful single material. We do this mass spectrometry, but you see this case, we have some noise, some other proteins together with our. And when we try to sequence this material, was not working because we have too much noise. So the noise, I will go back one step, noise needs to be very low in order to have the good option to do the amino acid sequence. Okay, then we try a different bacteriocins to purify. We use the H2C. And in this case, material that was giving a good activity was this one. The problem of this peak is not pure, because you have contamination from all this neighboring material that's going. But again, you run this one 5, 6 times, you collect all of them, these peaks together, you re-inject, and you hope to have it a single one. The problem previous methods, you have here, you can see it's a kind of extra material. And then we tried to do the mass spectrometry, was very disappointed because it was not pure. So we needed again to change the gradient. Who worked with the chromatography here? No one? Okay. Chromatography is not a bad technique, but it really needs to play around. So once you have something like that, you're happy because look like a single material. But when you do the mass spectrometry, it's showing that it's not. So we needed to go back, make a gradient much more longer to separate this actually. There was one peak on the front of the other one. And then you can even change the column. You can change the gradient. You can play on the gradient, but you need to separate because going to the mass spectrometry, you need to be sure what you have only one. And once mass spectrometry confirming that you have the pure material, you go for the amino acid sequence. But we was trying as well to optimize this process because obviously you're looking to reduce the junk proteins. Well, to grow the bacteria, you need MRS having the tractone, having the yeast extract, having the meat extract is a very rich environment. And obviously you need to reduce all these extra proteins to focus on your own protein. So we was performing one idea. Remember when I was showing the mode of action in the beginning yesterday, I was telling that sometimes bacteria can recognize proteins from listeria and use it as an inductor factor to produce the bacteria. So we was using actually MRS. We grow first listeria in the MRS, then we kill this listeria, and then we use this same spread media to grow our strain. But now we're having induction factor inside the media, and we presume that will be more bacteriocin produced. Then we do the ammonium sulfate precipitation against that pack, then do the size exclusion chromatography, and we was detecting. So in first figure, you're having Lactobacillus plantarum growing the normal MRS. This is the profile on the size exclusion. I mean, Himalaya mountains here. Here in between these lines is our protein. Everything's great. Obviously, we need to do more purification steps. When we first grow Listeria and then grow Plantarum, you see it's the same, more or less the same profile like here. They're not too different. Well, some difference, but again, it's a very high level of contamination of the area when we're detecting our protein. So, but interesting point, activity of the bacteria in here is like 10 times more if you first grow Listeria and then grow the Plantarum. So you have same contamination of the proteins, but you have much more activity. So it's already something we win from this extra stage. And then we decided to do another way. We took a MRS and we partially deproteinated with the amberlite. It's a kind of resin that is using in the purification process. So actually we reduced proteins in the MRS, and we observed that as well the strain can grow very well, can produce reasonably well, but again you see the comparing the profile of the protein profiles is much more less proteins around our bacteria. So it will be much more easy to take it from here, repeat several times, put a fraction that of interest, and it will be much more easy to have it, our protein recover as a pure protein. As I mentioned in the previous slides, we use the two techniques. We first grow Listeria to have the induction factor, or we deproteinated the media. And we observed that this could be a good approach to have a better purification and better production steps. Simple optimization of the bacteria in production can be done because MRS is not a magic media. MRS is a good media for growing lactic acid bacteria. But obviously for the bacteria in production, sometimes we don't need optimal growth. We need optimal condition for the production. We don't care for the growing the bacteria. We care the bacteria to be happy and to produce the bacteria. So we can play with some environmental factors in the growing and production. So we can do the effect of the temperature. We can grow the bacteria in 20, 30, 37, 40 degrees. Don't make sense to grow at 10 because they're not going to grow. I mean, at the Rohtak, 15 is already limit. Lactic acid bacteria like Lactobacilli, 20 it's like a more or less limit. But if you do 20, 30, 37, 40, maybe they will grow a little bit longer. It takes time longer to produce, but maybe they will produce better. We need to check. And as well, this is important experiment to know if the strain will serve poultry, because if it's 40 degrees, will serve poultry. Because sometimes you change 2-3 degrees Celsius and they're losing production. Because the control of the expression of the bacteria in operon, very often it's related with the temperature control. You're playing with the temperature, you can maybe lose completely your bacteria in production. Then effect of the pH. Bacteria can grow, lactic acid bacteria, they can grow in the lower pH, but as well they will produce. In this acid condition, operons will be expressed. So please check on the simple test. Just correct the pH of the growth media, 4, 5, 6, 7, 8, 9 maybe, and see if they grow and if they produce. As well, this important test to know if the strain can be applied as a probiotic, because when it's coming in the gastrointestinal tract, remember the pH is changing a lot. The bacteria need to survive in pH. How much is the pH of the stomach? Human stomach. 2 point something. Even can go 1 point something. So bacteria need to survive. We don't want them to grow in the stomach. They need to survive on this pH. Then when they go to the lower part of the gastrointestinal part, the pH going to grow from after stomach going up to 7. But then you need to be sure that in this condition, bacteria can grow and can produce. Commercial media. Okay, MRS, it's a very good option to grow the lactic acid bacteria. But Professor Santos will be very upset if you're using too much MRS media because it's expensive. So if you can have a different media that is cheaper and is producing, bacteria grow and produce, it's even better. So let's check different commercial media and see maybe some other media will be a good option. We need to check. Every bacteria is different. Every bacteria is different. And if there is no recipe, we know that certain media support the growth of lactic acid bacteria, so let's need to check and see which one of them will be appropriate. And then it's coming a point. Are you going to produce industrially bacteria in an MRS? Not going to work. In the industry, how much costs a box of MRS here in India? More or less. It's about maybe
5,000 for
$100, more or less, right? Well, one box of MRS media gives you about 10 liters of media. 500 grams of MRS is giving about 10 liters. In the industrial conditions, no way. We need to look for some waste products from the industry, and this coming point of sustainability. Because you're having several productions around the world that generate too much waste, industrial waste. If we can use this industrial waste as a base to create our own industrial media for production of the bacteria, it is the best option. There are several cases they're using cheese whey for production of the bacterial cultures, because cheese whey have enough nutrients and could be supplemented with additional some salt, some extra vitamins, but it's much more cheaper than use the MRS. As well, what else? Corn liquor, production of the sugar cane, it's related to this molasses production that in some cases we want to do. It's an environmental problem. And you can take this waste of the industry and you can actually produce, make your own media, use it for the production of biotechnological applications. We're having a project in the moment in Brazil using the waste from the orange juice production. I have not sure how many thousand kilos, actually tons, metric tons, you have every year waste of the production of orange juice. And these materials can be used for several biotechnological applications. There was an interesting project using banana peels to make filters for purification of the agua in the river, water in the rivers. So it's question, use your imagination, very often look in the traditional approaches. In small villages, you will be amazed to see how the people are applying all this waste product from the agricultural practices in the very innovative way. No one knows that actually they keeping in their hands a real treasure, because this one implemented on the industrial level is really solving ecological problems and generating additional profit, because using a very low, cheap material, primary material, that is even considered a waste, and you're generating a high level, high expensive, very expensive final products. So constant liquor, molasses, whey proteins, residues from the fruit production and treatment could be a good option. By the way, who like to go to gym? I'm allergic, I'm not going there, but most of the people taking whey protein, right? Do you know what was using the whey protein 50 years, 60 years ago? Giving to the pigs to have a whey with the muscles. So now we decided that we're not going to give it anymore to the pigs, we're giving to the people in the gym to make more muscles. But the principle is the same. You see, someone had a good idea. Observe it what the farmers do. Okay, this primary material can work well. Let's invest a little bit, and we're having a highly expensive products that we can sell to these people. Okay, but again, you cannot make your own media, but as well, if it's not to work, we don't have choice. So we need to work around the MRS and see how we can change the composition of the MRS media, because obviously some of the components we don't need it. You see, when we use the amber light with the proteinated amino, we have the same production. We don't need all the proteins. So you can play with the component of the MRS media or another media that you decided that is appropriate for the production of the bacteria. So you can play of type and quantity of the sugars. Remember, not all the bacteria like glucose, some like fructose, some like lactose. why you had API 50 CHL to know which sugars your strain can metabolize. And you can play and you can use it appropriately replacement on glucose with the media with a different sugar that will give the good growth and good production. Organic nitrogen sources. You can play with this. You have meat extract, you have yeast extract, you have the peptone, but just play with them. Just put one of them, don't put three. Put combination of two and play with the quantity, and you will be really amazed to see how this changed the production of the bacteriaceae. I was having a chat with some of you, and according to the regulations in India, you need to have 2 papers at least to go for the defensive PhD. Well, this optimization thing is giving a paper, and it's not so difficult. It's not associated with so much investment. Well, it's a kind of work in the lab for 2-3 months. You need to work, but well, paper is not coming as a gift from somewhere. You need to be generated, but it's again solving 2 problems. You can play with the quantity of the potassium sulfate, potassium hydrogen phosphate. Because in the MRS media, we're having only one of them, but you can replace. If you put a combination of them, you have the buffering effect. And this buffering effect actually can, in some cases, can increase the production of the bacteriaceae. Amount and the presence of triammonium citrate, part of the MRS media. So, well, we have several bacteria, and you don't need it, this triammonium sulfate. You completely cut out of the media, and you can have it. Manganese or magnesium sulfate as well. They're microelements, important for the growth, but in some time, bacteria producing much more better if it's in the stress, not in the perfect condition. And MRS was generated as a media as a perfect condition. So you're generating stress by removing some of them or increasing, and you have a better production. Production of presence of twin, it's a surfactant. In some cases, it's actually killing the production of the bacteriaceae. So in some cases, it's better to remove it. But as well, it's important because if you do the HPLC purification of your bacteriaceae, presence of the twin 80 can actually interfere with the column and can give you a hard time in the purification process. Presence of glycerol. Glycerol is not part of the media, but remember, you cryoprotected your strain in the presence of glycerol. Very often, taking out of the cryotube, you need to renew your strain 2-3 times to be rid of the glycerol because it's osmotic problem that can generate for the strain, and these osmotic problems can interfere with the production. Some vitamins, lactic acid bacteria, some of them, they need the vitamins for their growth and multiplication. However, some of them can have a positive or negative effect on bacteriaceae production, including the initial pH of the media can interfere with these processes. So you have your bacteriaceae, you know that bacteriaceae is produced, you know about the structure and all these things, but you know what is the genetic structure of your bacteriaceae. Interesting point, most of the bacteriaceae having some conservative and some variable determinants inside their genetic structure of the operon. And interestingly, ABC transporter is very conservative. Most of the bacteria, and you have the same ABC transporter as a part of the operon, that will be very conservative. It can be actually applied in very well, let me see, okay. So you have the yellow part that is transporter genes, and they're very conservative. So even if you have a completely new bacteria, the chance to have this part here, same one, it's a very high. So you can sequence, in fact, you have the completely new bacteria, you know, detected no primers that work to detect non-bacteria. You purified, you know the amino acid structure, you can predict potential gene organization based on the amino acid structure as well. But it's important to have the real one. You're starting from here, from the conservative part. And then you can do as called step out PCR. So you do the PCR that not going in the same directions to have the fragment, but you're going on the opposite direction. So when you do this PCR, you're starting from the conservative part and you read a little bit on this side and a little bit on this side. You have the PCR product, you sequence. Based on this sequence, you generate a new primers. Then start reading from here, and from here on the other direction. And after a couple of processes, couple of new primers, you will be able to reconstruct the entire operon. It's time-consuming, it's costly, but in some cases, no option. Well, you can always do the full genome sequence or sequence of your primer, your plasmid, sorry, and you detect conservative part based on your sequence, and then based on this conservative part, you can take a larger part, and you can extract from the sequence that you have from the full genome, you can extract the sequence of your operon. Because, these days we don't have option. If you want to publish a good paper, you're supposed to get some biomolecular work on the paper, and some biochemical, and some physiological, and some applications, so it need to be complex. No option, but again, it's working. When we was exploring, by the way, please let me know when I need to finish again. Because sometimes if you don't interrupt me, I can go up to 5 o'clock. And then I'm sure, why you took my time for the practical work? Will complain with me. Okay, in this case, we was using the electrode that can detect the level of potassium. So potassium and sodium, which one is inside the cell, which one is outside the cell?
Outside.
Inside is which one? Potassium or sodium? Potassium, right. So if you can detect level of potassium outside and inside, you can actually see if the cells was destroyed. Because if you're not detecting presence of potassium in the cell environment, this means cell is intact. In this case, we convert the results, and it was showing how we was detecting outside, but then we recalculate, and it was showing how the potassium inside the cells is disappearing in 1.5 hours of the different temperature in the presence of the bacteriocin, just using the simple electrode that measure the level of potassium. And you see when the Inococcus uni was exposed to the Pediosin PA1, potassium really in 1.5 hours disappear. But the important thing, you see that the temperature, 8, 15, 30 degrees Celsius. Okay, 30 it's work much more quicker. But since the wine production, when this trace is used, it's keep on cold, so it's important to know a date. But with this way, you have a good visual proof that, in fact, the bacteriocin was destroying the target microbes. Another method to do this modification evaluation is to find some molecules that are inside the cells or outside the cells and see if you can detect them. Okay, if you have beta-galactosidase enzyme that normally is present inside the cells, his job is there, inside, because he needs to break lactose to galactose and glucose. Okay, you have the galactose and lactose, so it's sugar, and then you're having in the effect of the galactose, beta-galactose will transform to galactose and glucose. The interesting thing, this beta-galactosidase enzyme is inside. So if we destroy the cells, we can detect higher level of the beta-galactosidase outside the cells. Okay, we can use the ONPG substrate that in the presence of the beta-galactosidase will get the product galactose orthonitrophenol. Orthonitrophenol is very important in this point because at 420 he have the yellow color, so you can detect in spectrophotometry. So obviously beta-galactosidase have his role, physiological and biological role for the bacteria. But when we expose ONPG on the effect of this enzyme, we can detect the product that is easy to be detected spectrophotometry. So you have your supernatant, you treat the cells of what you want to kill, you treat with the enzyme, and if the cell is killed, presence of enzyme will be fast. So you put the ONPG, and if you have transformation and you detect this yellow color, bingo, you have cells dead. This is a good way to detection of the effect of the bacteriocin by the simple method. You can do even more easy. because DNA, RNA, proteins, they're supposed to be inside the cells. Their job is to be there. If you destroy the cells by the bacteriocins, you can detect the increase of the DNA, RNA, protein on 260 nanometers. And here you can detect the presence of the beta-galactosidase on 420 nanometers. Only important point in this kind of work, you need to implement bacteriocin, they don't have a color. If you just take a simple cell-free supernatant have color because of the MRS. You cannot detect 260 nanometers on the something that is with this color. So you need to use semi-purified bacteriocin that is no color, and then you can do this method, simple method and detect that bacteria is destroyed because you detected intracellular material. Oh, very simple. We already talked about this. You just grow the cells, you put the bacteria in, and you observe the changes in the optical density. Or you can just, even more simple, you take a cell suspension, you expose to the bacteria and do the cell count before and after. This is a simple microbiological method, but method that 100% showing that you are able to kill your microorganisms. You can use it atomic force microscopy. It's someone already have experience with this technique? Okay. Normal electronic microscopy having a problem because you need to kill the cells, you need to coat them with gold or silver or other metals. In this case, you have the live cells, you don't need to kill them. And then you have the laser, it's coming and touching the surface and making something like I'm doing with my fingers here, making topographic image of your surface. And then based on the software that you're having, you can actually you can visually see that the cells are cut in some places and they're losing intracellular material. If you have patient, you can do several images one after one in the next, let's say, half an hour, and you can actually make it even video, how the bacteria go and really cutting the cells and liberating intracellular material. Size of the equipment is very small. It's about size of one laptop, more or less. Again, it's not cheap, but the problem needs to be in the place there is no vibration. Because imagine I have the microscope here on the table. I just come and touch the table, you lose the image. It's a very sensitive. Normally, we need to put it in the underground place with a solid concrete table to be no movement. And imagine even there was a joke, there was in one university was passing metro nearby. They say, no, we cannot. Because vibration for the metro, you don't feel it, but the equipment is feeling this vibration. There's some other images of atomic force microscopy when you have the healthy cells not treated and cells treated with the bacteria. You see how it's losing material from inside. The bacteria treated look like a cut it with a knife, some of them. So it's a very good approach to have these effects. Starting with the bacteriocins, they need to attach to the surface in order to having effect on the cells. I mean, they need to recognize the receptor, but this attachment could be interfered with the several factors. Could be temperature, could be chemicals, medicaments, pH can interfere. In this way, since they interfere with the attachment of the bacteriocin to the cell of the target cells, obviously you're going to interfere with their activity, their efficacy. We was performing the different tests, see what's the attachment, but again, attachment have nothing with this will work. Can attach very well, but cannot kill. So they're two different things. So we have this bacteriocin from Lactococcus lactis, and okay, its activity, it's active against Enterococcus fecalis, and have 60% attachment ability. In another case, 20% attachment but no activity. We're looking for some that is with, here. Streptococcus caprinus, 60% again absorption, like in this case, but no activity. Attachment don't mean activity, but to have the activity, you need to have attachment. And this percentage is very easy to determine because you have your cell suspension, then you want to kill, you have the bacteriocin, you know what is the activity before, You do the test, you do the time of contact, you measure the activity again, you calculate the simple mathematics. Interestingly, when you do the effect of pH, you can see that 100% absorption to this drain at 2 and 4. But then you go to the pH higher, absorption reduces. Why this is important? Because you see it's also in the temperature. At 4 and 10 degrees Celsius, 100%, 25, 30, 37, 80%. Why this is important? Are you going to apply a bactericide in the medical application? You're going to apply it as a part of the probiotic. You need to have a good attachment at 37. You're going to apply it in the stomach. You need to be a pH around neutral. You're going to apply on your skin as a medical application of the bactericide to treat some skin infection. Need to be related to the pH of the skin. Are you going to apply it in the fermented food products in dahi? So activity needs to be, attachment needs to be high in 4 pH. If you apply it in the food product, remember food product will stay in the cold, in the refrigeration. Obviously, you want to have a good attachment to 10 or 4 degrees Celsius. If you're applying it in the human body for some medical application, you want a good attachment at 37. In this way, you can actually predict where your bactericide can work and will work. Because obviously, this one that with a good attachment at pH 4, not going to work. And if it's having a good pH temperature attachment only at 4 degrees, it's not going to work in the human, but it will work good in the fermented food products. So based on, again, crossing your results of the attachment of the bacteria in the temperature, pH, you can actually go and predict, okay, this will work on medical, this will work on fermented food. If you have the fermented food products that is very popular here, fermentation made by bacillus, pH more higher will be good for this kind. You can check some chemical, how they affecting, because again, dairy product, presence of milk protein will be not affecting or improving attachment. Obviously, some of the examples are purely academic, because obviously I don't want to have methanol in your food product because you will be dead if consuming methanol. But again, some of these chemicals are important from the academic point of view, some of them even from the industrial point of view, because waste stream. Triton. Triton, it's used in the production of industrial production of the mayonnaise. So this can be important point to know. How long I have more? Because if you want, we can stop here and talk about safety on the next time, or we can start slightly and.
You have 10 to 15 minutes.
Can we make some kind of discussion now and to see, because rather stop to safety. Safety will go to the next one, and then we can discuss and see what's your question, what's your doubts, and what do you understand, what you don't understand, what you want to be clarified. Can you do it in this way? Okay. What's your question? If you don't have question, I'm starting to ask. Okay, what kind of techniques are you using here for the identification? Who work in the lab? Need to answer.
For purification, sir. For purification, sir. So we do purification. Firstly, collect the sulfuric supernatant, then go for ammonium sulfate precipitation, and after that, basically, we purify the cationic bacteriocin of probiotics. So we use cationic change chromatography. Then for removal of salt, we use gel filtration, and further confirm using SDS pH and HPLC.
And how do you happy with the yield?
Actually,
Yield is quite low, I know.
yes, sir.
But
Actually, the
this is academic exercise, and you wanted to know the structure, you want to know the pure material. From the industrial application, obviously not going to work, but I mean, this is different. Industrial and then laboratory things, sir.
Yes, sir. Basically, this is the main problem. Main problem is yield, sir.
It's quite low.
Yes, sir. Yield is about like 1 or 2%.
Oof.
Yes, Yes.
No, more or less, is this what you detected in most of the protocols? Nothing unusual.
Yes.
In the literature, it's more or less this you can have it. But have you tried the combination with to try to increase the production before?
Sir, actually, one of my colleagues is doing like optimization of bacterials in production using different like pH conditions. She optimized in medium also, named YGT. It has like cost effective, multiple times cheaper than MRS medium, and also use wastage of like agriculture waste, wheat straw and all. But till now, yield problem is still like the same.
Pushpa, can you go to see, can
Yes.
tell the optimization conditions? Is Pushpa there?
Yes, sir.
Why you are not hiding?
I was telling you not supposed to
What
afraid.
matrix you are using for telling the optimization condition he is asking?
I'm optimizing the condition in SSF and solid state fermentation and at submerged fermentation as well. But vegetative in liquid medium, the growth and optimization is easy, but in solid state it is quite difficult. But sir, liquid purification of bacteriocin is done by liquid medium. Vegetative, I had named that a vegetative medium.
Actually, the terrible point is some bacteria they don't want to produce in the liquid medium. Sometimes they produce it only in the solid fermentation.
Yes.
And when you have the solid fermentation, okay, I have good activity, but then how we can extract it?
Yes.
There's coming another problem.
Yes.
You see, sometimes the nature don't want to collaborate too much. They're making our life difficult. But this, I put this image of Mona Lisa because it's art. You need to try to explore and to find a way. There's no other option. And your supervisor wants the results, and he wants a paper in that. It wants a good paper as well.
No, actually she planned that experiment on the solid state because most of the application is on the solid.
Yeah,
We grow in the lab because of our convenience in the broth.
yeah.
But in the food and on the gut, if you go for application, the organism grow on the solid surfaces. So she put this objective in the thesis, the aim that we have to optimize this condition on the solid surface.
Yeah. To be more
So she
real.
took like a wheat flour and wheat bran and
Rice plants,
so she
more
has
plants,
optimized.
different type of plants and different type of biowaste, sugarcane, bagasse, congress grass, coffee, many type of solid states.
Yeah, so whatever we have agriculture residue, so there are two things that we are valorizing this waste to gold, you can say, that by growing these organisms. She found that problem that when you grow on the solid surface, the organism is growing fantastically because we cannot see the body. So you have to go for CFU per ml and calculate the viral cell. The growth is fine, but when you extract, you get nothing.
Yeah.
Because whatever is secreted by the organism is absorbed by the matrix.
Yeah.
Mostly because of the hydrophobic condition. Most of the bacteria, especially, they are hydrophobic. So once they absorb, you are not getting that. So still, we were encouraged that organism is growing at least. We have tested that it is a producer. But when it is growing, probably it is producing. But yes, of course, that problem is to how to extract growing on the substrate. So you have to
Very
go
often,
in the broth.
the models that we use very often they're designed for the liquid media. Very often the models, and then but this is not realistic always because as
Yeah.
I say,
Yes.
well in the tube it's going very well, but when you go to the real application there's nothing.
Yeah.
So it's really needs to find the condition and to optimize and to go
Even
deeper.
even in the broth medium, you see the MRS is a highly dense dark color. So there are so many impurity peptides are already present there in the medium.
Yeah.
And if
Sometimes I call this coffee color. Look like a coffee.
yeah, it's a typically Indian tea color, like you dip with the color becomes the same like the supernatant color we get after the growth of the
And very often,
And she did that one also to remove the conditions by one the all the MRS medium. So one by one she was trying that which one is producing this color. By doing that, she optimized at least minimum level of these nutrients and producing the equal amount of bacteria in the broth. And now how much cost you have reduced? The MRS, if you compare with the MRS and the
Three times
medium you
cost is
optimize,
about 3 times from like less than from MR.
and she has developed a medium 3 times cheaper than the MRS, but it is producing equal amount of weight.
Yeah. But there's another problem. No one in the industry will use the MRS for the industrial production. It's extremely expensive.
Yeah.
So, and then again, we need to go back to this step that we can use the waste products. Because if you, again, sustainability, you're removing the waste. I mean, another curiosity. Do you like cashew juice? It's popular here in India.
Yes.
In Brazil, it's very popular. Cashew nuts. They're making juice from the fruit. There was a curiosity they told me a few years ago why they stopped producing this juice. Because everyone was interested in the nut. And they was having a huge residue problem of the fruit himself. Because there was no value market for this fruit. They tried to give it to the animals. The animals not like it too much. And the problem is you have high level of tannins. And even if you give to the animals that you are producing milk, you can even test some tannins in the milk later. So obviously it's not working. So they went and they made this kind of modification, and they was making juice that was reducing the tannins by the biotechnological approach, and they was producing juice. So you see, one the waste that was waste become a highly valuable and expensive juice product. So I think it's another option. We always need to look into optimizing condition, how we can use the waste product from the agricultural industry.
And Brazil is the largest producer of sugarcane.
Yeah.
And the sugarcane bagasse is one of the problem. Maybe
Yeah.
what we
It
do.
is. It is
And-
huge problem.
And if we are able to utilize those bagasse for the production of this application bacteria, even at the cellular level, it's not the metabolite, even the straight.
Okay, I know that its lecture is recorded, but I just cannot hesitate to give this example. Everyone climbing these days, the electrical car, it's a very good. It is. It's less pollution. It's true. I mean, in the city, it's much more less pollution. But someone talking about the people that are involved in the production of the metals, mining, to produce these batteries. So in fact, pollution was moved to another place where is production of these metals for production of these batteries. So when we talk about sustainability, we need to look at the entire system, not just the city where we live in. Because for me, living in the big city, having electric car is excellent. It's my air is cleaner. But we need to think about the people that are involved in the production of this mining industry. They're producing these metals. Very often they have a cancer, they have a very difficult life. Very often this is how they call third world countries that no one cares for them, for their health. We need to care for everyone because in the end of the day, if one part of the planet is struggle, you struggle entire planet. You don't have regional things. You need to think globally and to be respecting everyone. So this is the point. I mean, people love to have cashew nuts, but I mean, all this residue that is produced and been wasted in the past needed to be used and need to be explored and to be producing a high value level products from this. So every waste in our perspective needs to see how we can apply it. And biotechnology is a good option because we can use this waste product as a substrate for the production of the other products. Because we don't have second planets to move there. We need to keep it one that we have. I hope the car industry not going to hate me for this comment, but again, we need to look for these people that are involved in this production as well.
You can take the question from participants. See,
Jason, if you don't ask a question, I will ask difficult questions in the test in the
my
end.
dear, see, you sit and go will not help you. This is opportunity for you. Whatever you have question, it's not high-fi what is talking, what me talking, okay, we are talking our research, but whatever basic question you have, come on with that. Is it just coming and eating pakoda and going? I'm not against of eating pakoda. You can enjoy the way you want, but we should have pre-plan your question.
And don't be
अभी
shy.
थोड़ा जल्दी में लगता है ना कि ये कुछ बोल रहे हैं, मुझे क्वेश्चन तो आ ही नहीं रहा, पूछूं क्या, ऐसा होता है। घर से आओ तो एक क्वेश्चन हर बच्चा लिख के आए, लिख के आओ कि मुझे ये पूछना ही है, ठीक है, इस फील्ड से नहीं भी हो सकता, हो भी सकता, लेकिन ये मेरा क्वेश्चन, ब्रॉडर एरिया तो यही रहेगा ना। आप एक क्वेश्चन लिख के आओ घर से, 40 बच्चे हैं, आज नहीं कल, कल नहीं परसों, आप इनको 5 दिन तक कुछ ना कुछ जरूर पूछो। I'm just motivating them to ask the question, whatever they want, and write down from their own, and then ask either today or tomorrow. Not a problem, because if there are 40 and take one question each, you'll be happy to answer. If not today, then tomorrow and next day. That will be better.
Another thing to remember, I was telling you this example of chiseling. In the beginning, like 40s, 50s, 60s, the last century, they was giving to the pigs to have growing pigs bigger and then cut the pigs and have a party. But again, I was using this one. So take one. So someone have this idea. Let's make this product kind of concentrate, better purified, and offering to the people in the gym and making lots of money. Sometimes simple ideas can be a very good option. And I've always been criticizing that I'm looking in the small villages, but small villages giving excellent ideas. What you can use. Plants used for the medical purposes, you just find what molecule is there, it's important. Or look on the fermented food products, why bacteria there is so important. Why the dahi from one village is work better not in the other village, because they have cultures that established there. As a starter over the several generations, there was just natural selection of some good strains.
It's a very good idea he's telling. It's not that a high-fi publication I go and do research. Go to your village and find out the exact problem what you have and contribute that how I can solve. For example, in Haryana we have Paradi problem. This the residue of this wheat and rice, they burn in the field. These farmers have no option, so they burn it. And it's a huge problem to the pollution.
Okay.
Sorry, I will take a minute only. In Delhi or nearby area, the government is notifying so many instructions that hey, don't do this. It's a problematic, asthmatic. A lot of people are facing several issues by burning. And second, it makes the soil sterile because you are killing all the microbes in the soil which are helpful for the growth of the crops in the next time. If you burn that everything, your soil will become completely sterile. It's not fertile now. So there's several problems. Just one or two. Go to the village. Can you use this wheat straw? The farmer will give you free of cost. You are not going to pay it. And your bacterium, what we were talking about probiotics, you can grow the probiotics using just your wheat bran or rice bran or sugarcane bagasse or whatever the major problematic residue, you can select it. And we are using this as a medium. People have used MRS or very, very refined medium for the growth, and it is a very costly. And now you go to the government, say, I have an option. Don't use this MRS because it's very, very costly. I am using a product which is a residue. The farmers are throwing this. Catch this project, government will happy to announce you the fund. They will give you a grant and they will give you a job.
And after you make your own product.
They will give you a job that you work on this topic throughout your life. This is the transforming period some of the people have faced and they got it. Really is inspiring. I'm sorry.
No. When you say the rice production, okay, now I have a problem. I'm not supposed to give the details because it's under patent preparation in the moment. But with some colleagues, we're using the extract from this waste from the rice production. So that this external part of the rice, extracting some parts and making actually sunblock creams on this. That is good for the skin and protection for the UV. You see something that's considered waste, and as you say, they needed to burn because it's a problem. But we're extracting some piece of molecules. I don't even supposed to say what kind of these molecules, and these molecules getting part with the good antioxidant properties and a part of the composition of the cream for the protection of the skin. But you can go. I mean, I was working in Africa. You see how they're using some plants there and make a kind of powder of these plants and use it as a sunblock. It's a traditional knowledge. I'm sure if you go here with small villages, you can find this kind of traditional recipes for several products. And you just go deeper and you discover what is the molecule, what is the bacteria involved. And again, you can make a product, you can have a patent, you can have a projects, you can even have some of the patents can give you really good money. Okay, money is always motivating, and knowledge and curiosity.
Yeah, so this is how my suggestion: make the way you want to interact, the way you ask the question would be helpful. Just sitting and listening is one way of understanding the concept, but sometimes your speaker flows on his own way, own pace, own thoughts and knowledge. But you should have that way of interact to explore better the way you can. So thank you very much. Yes, sir, please.
You had mentioned the industrial biowaste from sugarcane. Molasses, I think you have been working on two bacteriocides. Are they in some patent stage or where that has?
No, we don't have patents on using the sugarcane as a growth media because was well, we publish results. We show that we can use the sugarcane molasses as a component, principal component of the media. And we was like stating, okay, we can use it, but no patent, and it's something that could be explored deeper and applied. There was even using for production of additional levels of the bioethanol now, some works, because it's, as Santosh mentioned, in not only Brazil, all around the world, this is a big problem, a residue problem. And again, if we can manage to use it for something, it's reducing the environmental problem. We're not going to solve the environmental problem, but reducing one step, reducing another step is always a good direction. And you're producing something with the cheap substrate. Could be bactericide, could be, even if you can grow some bacillus, can even be antibiotics, can be enzymes, can be used for the several applications because it's quite a rich substrate.
Thank you.
Like there's a take-home message is you have to identify your residue from the kitchen to the farm. You throw everything. It's my dear student, you are throwing a gold. Why you are throwing? Because you don't know. Once you know, you are discovered. Since dawn of the civilization, people are throwing. Those who comes in the way, okay, don't throw. This is how we can do. They are the discovered. They are known by their name since several years. So this is how we have to identify. This idea, small idea comes into a big picture and then can help. So go to home, learn that what these residues are, what is their property and how we can utilize this also.
Grandparents always giving a good idea, right?
Always you can get idea by discussing with him, not just I'm again lecturing because you are part of the program. You listen whatever, but after the lecture, it should be entertaining. So this is my request to all the participants. Our program is live webcast to the Ministry of Education. We cannot compromise with the timing. So come on the time at 10 o'clock starts and finish up to lunchtime, not a problem. The delay is not a problem, but you start at least on the time. That is okay. And try, don't switch off this light because the speaker is in the dark. So
But I asked actually to switch it to see
while,
at the screen.
so while the light webcast, there was a dark, nobody was visible. So the phone call came to me that your program is running, but we cannot see the speaker and the participants. So I think you can see the slide in this, but we'll try to switch off some of the light if not all, so that you should make a balance is visible because it should be there and the recording mode. It's just a condition for the continuous. It should be a live webcast with the recording. But thank you very much for your interaction and listening. We will be sought back at the Department of Genetics in the lab. Right? Thank you very much. Pushpa?